after And so after 5 days and 5 nights of hacking or so, approximately, Neuro Academy is coming to an end very soon. But before it does end, we're here gathered to hear about all the wonderful things that you have done over the last few days. And so I've posted in Slack a link to a Google Doc for you to put in like run of show, the order in which we'll go, and we'll sort of crawl on you one by one. The HackQ tab, which I believe is now called something else already, team have asked to go first. What we'll do is everyone, every time you come up here, actually I'll demonstrate this. Every time you come up here, you can put your laptop here. You'll need to share your screen, or someone on Zoom will share their screen from their side to present. And then present. We'll ask presentations to be 10 minutes or less. And again, just to emphasize, if you're here in the room and you stand here, then the owl can see you, then people on Zoom can see you, and then the recording will also have your face rather than your back. So stand all the way back here while presenting, and then go here to switch your slides or have somebody else switch your slides for you. All right, and we will start with MLP tryout. Woo!
Hold on. You have to join the Zoom, sorry. Yeah. I am on the Zoom. Where's my screen? On this one? No. And then it should be both screens. Because right? But how come I can't share this one? It's gone. Okay. And then I go back. So it started the screen. Yeah. Can you see it? Oh no, you can't see it. Can you see it now? Okay. Good. Okay, hello. We are Team MLP Tryout. Just to give you an introduction of who we are, we're a really global team from all over the world, different areas of expertise and different time zones. So it's been a really interesting experience working with everyone. We have Raj, who's in deep learning, Selin, who's in clinical psych, Woonju, who's in psych, I'm in neuroscience, and Maiwen in computer science, and Nadine in computational neuroscience. We use the Amsterdam Open MRI collection, and specifically this composes 3 data sets. And we use the ID 1000 data set, which consists of like 900 subjects, but we only ended up using a subset of that. It was around 16. We used functional data from fMRI scans involving movie watching. We also used T1 weighted data for anatomical data. And there were also some behavioral measures like the intelligence scores and the BIS-BAS behavioral questionnaires. And just to show you a bit of what the movies looked like, they were composed of naturalistic scenes. It was a continuous movie with music playing throughout. And there are some images. It was mostly landscapes, but there are some images of faces later on too.
And I don't think we were able to hear the sound, but in the movie, there was also, am I standing? Okay. There was also background music playing throughout the movie. So it didn't have any lyrics, didn't have any human voices in it, but musical instruments, with different pitches and some changes in tempo, the rhythms, depending on the scenes. So because we are in a Neuro Academy, we had technical goals, right? There were things that we wanted to learn and try. So we first wanted to work with a big data set. And then all of us wanted to try machine learning. So that was a huge goal. Thus the name MLP tryout, which ended up kind of irrelevant. And then third, we also wanted to understand the auditory processing and extract some auditory features from the stimulus. And then we derived the research goals out of the technical goals that we had. So first, we wanted to see if we can predict the auditory features from the stimulus from the functional connectivity. And then second, if we can predict the behavior, all the psychometric measures from the functional connectivity or the anatomical measures. So we really wanted to try exploratory approaches and see what we can do with this data set. Here's an overview of what we did. So first, we extracted auditory features from the stimulus. We parcellated selected ROIs and extracted bold time cores, and then made functional connectivity matrices and built machine learning models. In detail, so to extract auditory features from the movie, we used a Python package called the Pliers. It's a tool for analyzing music. And it basically gives us the normalized energy in 5 out of pitch classes. And because our stimulus was continuous, we used this sliding window technique to extract the partial chromogram in each of these sliding windows over time. Each of these windows were 22 seconds long, and the interval was about 4 seconds, 2 TRs. To parcellate, we use the what's called the Diffumo parcellation atlas. I think it's a dictionaries of functional modes. So this atlas has been determined based on the functional activities, and depending on how fine you want to go in terms of the number of parcels, it gives you different numbers from 64 to about 1,000. And we wanted to start simple, so we went with the 64 parcellations. And we selected also ROIs within the 64 parcellations to reduce the dimensions. We did some quick previous research review and came up with the auditory areas that are known to be selective to music, and primary auditory cortex, as well as the component 7, which is involved in song production as well, like in singing. And then within those ROIs, we extracted the bold time courses and made the functional connectivity matrices. And here we use the same sliding window technique across the time course. And this is showing the 64 by 64 functional connectivity matrix, which because it looks nicer. And within this middle region, that's where the auditory cortices are. So it shows nice activation, correlated activation among the auditory regions within this data set. Okay, now I'm going to pass it on to Raj, who is going to explain the machine learning models.
Hey guys. So I spoke about the what ML models we tried to use and explore as a first pass on the data. Next one. Next slide. Yeah. Yeah. So the first thing was like we basically tried out two ways of taking data input. One was the functional connectivity matrix, and the other one was the measurements of surface area, volume, and thickness, and so on. So we basically had two aims as mentioned before. So we wanted to basically look at audio features prediction from the anatomical and functional data. So we use basically 16 subjects in this approach with the window technique, and we also use the overall connectivity to to basically predict some of the behavioral measures. For example, we try to predict the intelligence structure test scores, and also the BIS and BAS scores that we had. So we tried like things like, for example, ridge regression, KNN regression, MLP regression, and support vector regression, and we to make sure that we don't have any sort of data leakage, we had this nested cross validation sort of an approach to make sure that we don't use the test data into our training data in some sense and get measures which are inflated, and we use grid search to choose the best hyperparameters for these algorithms. We also try to use permutation test to understand what correlation measures we are getting. Are they random, or can we assign some sort of a p-value to them and see if they are significant or not? Next slide. Yeah. So the future, we basically want to see, like, can we build multimodal models using more data on functional data, anatomical data, and we also have physiological data, that is the cardiac measurements and the breathing patterns of the subjects during the experiment. And we also have, some more tests that we could try to integrate into the models. We could also try to use CNNs directly on the bold and the team-weighted images. But as you know, like, CNNs require a lot of data. We also do have a lot of data, by the way. So we have like 1,000 subjects here. So we think that using CNNs might be really possible in this particular data set. We also have a we could also work on parcellations using NeuroQuery, because it already has, like, regions which tells you which parts of the brain get active for a particular task. So we could use these parcellations in conjunction with the parcellations that we already have from Diffumo and see what works better for our use case. And yeah, mainly take all subjects. We actually work with a very small subset of the data set. So the main aim would be to take all the subjects and look at how does the measures then change. And the main point being, like, can we make it reproducible, and can we solve all the issues which come with statistics in this particular field of neuroscience. Next slide. Yeah, so the most important part of what we've learned as part of these 2 weeks. So for me, like, I always had like clean data given to me, and then I used to work on building ML models from that. But this particular 2-week hackathon gave me an idea of like how to take the pre-process fMRI and bring that to ML algorithm. That particular pipeline became much more clearer, and I also got clarity on how we can make sure that our work is more reproducible, and how to deal with issues related to p-values and stuff like that. So it was a very big learning experience personally.
So yeah, the most important part. So I learned about basically, using Python and all these, packages to extract the auditory features, parcellate, extract bold time calls, course, or all that, and then also navigating big open data sets and the machine learning. And most importantly, I think I really enjoyed this process of collaboration, coming up with ideas and thinking about what we want to do all together, and, really collaborating with people in different time zones. So yeah, that was really a good experience.
Yeah, I definitely agree with Woonju on the last part. I learned about how, like, what the difference was between different types of parcellations. I learned about meta-analysis tools like NeuroQuery and NeuroSynth. I feel like I know a little bit more about machine learning now. And just to go with acknowledgements, we had a lot of help on this project. From Noah, who helped us a lot with troubleshooting, Molly, like, especially since our team was global, having those Zoom videos up like in a timely manner was so important, and it was so awesome. Satra directed us to the Pliers package. here has just been very helpful. A lot, and of course, Ariel. And thank you. Thank you all for listening.
Thanks a lot. And we will go on to the next team, which is the Paranoia team. Come on up. Paranormal events. So are you, oh yeah, you're presenting for.
We're all in person.
You're all here in person.
But I think,
His laptop is sharing.
yeah, I think he thought that was,
yeah.
is there a better way to do it?
No, that's the way to do it.
Okay. You start. Okay. Yeah. Cool. Okay. Wait, go back a slide. Sorry. So we are the Paranoia group, and essentially our goal was to explore the influence of traits and certain semantic content on neural event boundaries. But before that, let's actually go into what event boundaries are. So I guess if like you imagine you're waking up in the morning, you're in your bedroom and you move to the kitchen, like that shift from one location to another and the actions are discrete to those locations would probably have you segment those two things into two separate events that are separated by an event boundary. And typically, like experimentally, this is studied using continuous stimuli like movies or audio narratives. And then this video I think is really good because it kind of shows how like behaviorally we kind of chunk information into events. Neurally, you'll see like during a sentence or during an event, there's a consistent activity. And at some point, the sentence is going to change. And now this sentence kind of has a consistent activity, which gets at this idea they have stable neural activity during an event. And then there's a major shift to indicate the start of a new event. And the way that this has been studied has been, let me go to the next slide, using hidden Markov models. So essentially, if you look at voxel-wise activity, you're going to detect moments in time where there's a major change across voxels, and the model will basically identify those state changes and label those as an event boundary. And then typically what we get out of this model and what you'll see many figures of today is basically we get these time point by time point correlation matrices. And then on them, we can basically label these squares, which indicate activity within an event with models with these lines indicating these event boundaries.
So regarding the data set, we used open source data. In particular, we used data from the second session where subjects were listening to a paranoia-inducing story during an fMRI scan. We only used 17 data sets from the complete data set. The story was subdivided into 3 runs. And you can see also a segment of the story that induces paranoia that we extracted from the complete story. Previous research with this data set has shown that people with high paranoia levels showed similar neural activity. And so our goals for this project, we wanted to see if it was possible to replicate past findings in a new data set. And specifically, we wanted to explore the influence of traits and content stimulus content in individual differences in neural event segmentation.
So yeah, in prior work, well, what are the prior results that we're interested in replicating? In the same work that established the hidden Markov model as a technique of detecting event boundaries in neural data, the authors demonstrated that different regions encoded a different number of events based on their location in the neural information processing hierarchy, such that, for example, a region like the visual cortex encodes a higher number of events as that region's tracking lower level features of, in this case, a movie stimulus that are changing more rapidly, whereas a higher order region might be demonstrating an encoding of a lower number of events as it tracks a higher order feature of the stimulus. And so in order to investigate whether we could replicate this in our purely auditory stimulus, we hand selected some regions that have been shown to track event segmentation in previous literature, such as the posterior medial cortex, angular gyrus, medial prefrontal cortex, and the temporal pole. And using this, these hand-selected regions, we're able to demonstrate that, for example, in the MPFC, a relatively smaller number of events are encoded compared to the angular gyrus. And if we zoom out for a second to a whole brain parcellation, brighter, more intense red values here demonstrate a larger number of events being encoded. We can see some broad, rough structure of the same thing here, right? Where lower order regions are encoding a larger number of events and higher order regions are encoding a fewer number of events. Although in the analyses moving forward, we will be relying primarily on the hand-selected ROIs.
Okay, so our next question was basically we're trying to link semantic shifts in the stimulus. So for instance, like we're describing one scene and there's a move to the next scene, we're trying to relate those to neural event boundaries. And the way that we did that was we actually took the story text and then extracting from the story text, we used topic modeling and certain semantic, like universal encoder vectors and sentiment values, and we extracted all of these. And then we also use human annotated boundaries as a control because it's been shown that human annotated boundaries seem to correlate really well with neural event boundaries. And then what we did was we took those vectors, generated basically text similarity matrices, where again, time by time, you can start to see this like core structure across the diagonal. And then we use a hidden Markov model on these text matrices to identify events and compare those to those neural event boundaries to basically see, is there alignment in the text in terms of semantic shifts that aligns well with actual neural shifts in activity. And we end up finding that this is kind of the case. So to orient you again, this is just the vertical lines here, actually moments over time where there is an event boundary. So there's a shift in events. And you'll see again, kind of pointing to what John Andrew said, the angular gyrus has a lot of frequent events. And as you move up the hierarchy to the medial prefrontal cortex, there's less events. And you'll see that, however, like across the entire stimulus, there's moments of time where all the regions align, there's state changes across the entire cortex. And then importantly, I annotated the boundaries. I was our human annotator, and my boundaries align well with the medial prefrontal cortex as well, which kind of supports our hypotheses.
And so we can look at how these event segmentations might emerge over the sample overall, but we can also look at individual differences and how people might be chopping up a continuous narrative into discrete events. And one way we can do this is by looking at how individuals might differ from the group average event segmentation. So plotted here on the left with time point on the x-axis and expected event on the y, the dotted line demonstrates where do we expect subjects, what events do we expect subjects to be in at any given time point. And then on top of that, we can plot each individual subject's event timings to see are certain participants consistently delayed or lagged behind the average event timing or consistently ahead. And so if we visualize this per ROI and per run, we can see significant variability across, both ROI and run, although you can see in the MPFC and the temporal pole, there's some additional variation here. And if we quantify the variability here as the difference between each individual's timing and the group average, we can try to compare this to individual trait level paranoia. And we find that as paranoia score increases, so do subjects' divergent scores, suggesting that these subjects might be processing the continuous narrative in more idiosyncratic ways.
Okay, another thing we can do with this data set is we can look at how the individual differences in event segmentation is related to the characteristics of the stimulus. So by looking at that figure to the left, we can see that there's some time point where people seem to be pretty far away from the middle, like diagonal line, but there are some points when there's less divergence. And we can use that, we can calculate Jason-Shannon divergence based on this difference in time point, and we get the time point by time point, how different people are from each other in what event they're in. So we also get the sentiment analysis and topic change from the from analyzing the text, and we can overlay the sentiment analysis on top of the Jensen-Shannon divergence, so we can see how the sentiment change in the story might be related to how people process, how people segment their experience differently. We found a pretty mixed result that we don't trust too much, but basically there are a bunch of regions in some runs that shows positive correlation with the changing sentiment contents, such that yeah, when the sentiment is positive, people become more divergent, and when the sentiment is negative, people become more similar. And there are some regions that shows the opposite pattern, but like the results not robust. It's not consistently showing in all runs in RI, and like it's subject to some changing parameters. So we don't trust it too much. We need to spend way more time looking at stimulus to figure out what's going on here. But to conclude, as previously shown, event changes vary across the cortex and correlate with human annotation of event boundaries. And we found that sentiment and topics change throughout the stimulus, but we need to improve our sensitivity to detect these changes. We found there might be some relationship between individual differences in paranoia and the divergence, and there might be some relationship between stimulus characteristics of the divergence, but this is beyond the scope of a 1-week hackathon. So yeah, thank you. Also, it did exactly 10 minutes.
Nice job. That was great. We will next have one of the many teams who worked with the NSD dataset. I'm very excited to see all the NSD projects that grew this week. So next to NSD, BioSoc. All right. Oh yeah, you please join Zoom, and then just again to remind, when you're presenting, look at the camera. That's right there, okay? Instead of standing like this, like that. And then put your slides over here so you can look at them on the screen. Oh. You need to join. Oh yeah. I think there's a message with like the Zoom link is the most recent. Do you have the presentation also? Yeah. Okay, so yeah, as soon as you share your screen.
Recording in progress.
Right on. Whoa. Visual effects. Okay. Yeah, and just use that screen instead. That's
oh yeah. Okay. I think that we're good. Oh, right. Okay, thanks for bearing with us. Hello, I'm Avery, and our project title is Predicting Social versus Non-Social Natural Scenes from fMRI Responses. Does it work? I'll just do this. Oh, okay. I think I just had to click on Zoom. So this is our title introducing slide. So hi, I'm Avery again. I'm a grad student at the University of Iowa. I just finished my first year and I'm in the neuroscience program. Do you guys want to like come up individually?
Okay, I'm Madison. I'm a clinical psych student at the University of Nevada, Reno. I study trauma and individual differences.
My name is Marissa. I'm a rising third-year student at Penn State in developmental psychology.
My name is Veronica. I'm a PhD student in the bioengineering department here at the University of Washington, and I study encoding fear or anxiety.
I'm Erin. I'm a rising third year at Michigan State University in the psychology department, and I study skill acquisition and expertise.
I study social cognition. I didn't say that. So this is just a quick outline. We'll go over our project goal and then data set, imaging methods, computational methods, and then our discussion. So our broad project goal was to take the bold signal from these fMRI scans where they're viewing natural scene images and take the social and non-social images and see if we can like predict what image someone's viewing based on their bold activation. And then our like technical goals were to work with machine learning, some imaging data analysis, GitHub, Python, all that good stuff, big data. Yeah. Okay.
I'm good. So I won't bore you with all of the NSD stuff because I'm sure you're going to hear a ton of it today, and we heard a lot about it last week. So basically, this is the data set that we worked with was NSD. They got about 73,000 images from the COCO data set, which is an online available data set that has all of the images segmented and annotated. So that's what we used as our stimuli for this project. And then, so for annotations, we wanted to have multiple raters so that we could like reliably say that this is a social picture that they're looking at, or this is a non-social picture. So we had 3 raters. One of them was the COCO dataset annotations. So that was anything that they identified as a human body in the picture. Then we also had, someone else had pushed some like open source code for something called YOLO face, which basically did the segmentation, but only for faces so that it wasn't just like someone in the background of a picture. And then I was a human rater. So I went through all of these images that they used. We used the 1,000 shared images. That way we knew every single subject saw them. And I went through this and I basically looked for any human in a picture to deem it social versus non-social. That way we could do it like with certainty that someone could actually see it. That wasn't a computer. I lied, I'm gonna need that. So just to give you like a basic, this is how we coded things. So this is a social interaction between 2 people. This is a social interaction between 3 or more people. This is a non-social non-interaction with 1 person. All right, sorry, that's a social non-interaction with 1 person. This is a social non-interaction between 2 people. On that side, it's all social because there are people in the background, but there's no interaction and there's multiple people in a group. And then this is a non-social non-interaction, no people.
All right, so just a brief overview of what we did. We decided to use the MNI betas that they had already provided to us from the data set because we wanted to save time trying to figure out how to convert it from person space to MNI space later. But then of course, there's bigger data. And so one of our goals was to figure out how to work with big data. So what we did was we extracted the betas only for those shared 1,000 images because we had obviously one for every trial, but we wanted only the ones for the shared images. And then because it was so big, we had to downsample so that we had less data to feed into our machine learning algorithm. So we smoothed and then downsampled the data.
So then once our data was prepared, we had them in separate NumPy files that we had each split up to get and put on the AWS bucket. And then we collected them onto one of our local machines in the Jupyter Hub. And then we were loading them in to put that into our SVM. We decided to start with the simple model just to see how it performed. But when we were doing that, it was still too much memory consumption. So we decided to, instead of creating one SVM across all participants, we created a separate SVM for each participant across all of their sessions. And we didn't end up with very good results. It was pretty much around chance performance in predicting the social versus non-social images that were being viewed, except for one participant who performed around 75%. So that was interesting. We think that this might be because even though we did downsample and our data was much smaller than it was initially, we still had like over 100,000 features that were basically going into an SVM, and an SVM doesn't deal with this very well, especially with we still had a pretty limited number of like rows that were going into the model. So that's an area for potential future work for our group. And now I'll pass it off to you to talk about that.
Yeah, so like Erin just said, because our data was so large in this flattened array in the SVM, one way we thought we were going to try to approach this is to basically reduce the dimensionality of our data set by projecting into principal components and then projecting our data set onto those principal components and basically using a smaller number of features in this case. So that's one thing that we're actively working on. This was just a confusion matrix that we had for our best sample in the SVM. Okay, so for the next steps, our first approach is to look at subject to subject variability. So we were generating these SVM models for each of our subjects with the hopes that if we identified some features in subjects that allowed it to make these predictions or accurately predict the labels, that we could then go back and look at what these features actually corresponded to in the imaging data. So we're going to still try to assess some ways to do that. And then for future directions, as you've heard already, some of the other groups were looking at specific ROIs. We think that this is one way to effectively reduce the dimensionality of our data and be able to handle it more easily. So hopefully we would go back to literature, identify some ROIs that might be important for identifying social scenes, and then use these in our analysis. When we initially conceptualized this, we were thinking about using a convolutional neural network, and I still think this is a pretty good idea. We went back to try a simpler machine learning algorithm, but because the convolutional neural network could handle these three-dimensional input arrays that are going in for each sample, we think it's potentially a better model for the type of classification we're trying to do. So we've wrote some code to actually use a convolutional neural network, and we're in the stages of preparing the data to be input into that. And then as Madison talked about, one of the other future directions is, instead of just using the social, non-social labels, to incorporate a more specific data set that looks at interactions versus non-interactions. We learned a lot. Okay. So I don't endorse this, so somehow it's become a staple in our team is to use git force reset. Not sure this is the best practice. Again, I don't endorse it. And our favorite data structure is pandas data frame with NumPy arrays in the cells. So just kidding, don't ever do that. Yeah. Project info here.
All right, thank you so much. I think we have another NSD group now, the functional subbundles. Yeah, the pandas with the NumPy array in it, I've done that. Definitely. I don't recommend it, but I've done it.
Okay, hi everyone. I'm Stephen, and I'll be presenting on behalf of the functional subcomponent of bundles team. So as I just described, this FSUB acronym here stands for functional subcomponent of bundles. So essentially what the software is going to end up doing is taking white matter and defining or extracting white matter based on how it intersects with gray matter regions, such as those that might come from a functional ROI analysis, like a cluster that you might get from that. So before I describe how we do this, I want to start by acknowledging our team. That we have a very, for our team, we have a very diverse range of expertise and perspectives that made this whole project possible. So how it works is that we start with a functional ROI from fMRI data. In this case, we used the face fusiform area from the natural scene data set. You can see right here. And the code has basically two different methods. The first is that if you only pass in one ROI, such as how we defined here, it'll ultimately find all streamlines that intersect with that ROI. And if you pass in two, it'll find all streamlines that connect the two ROIs. So for now, I'll just be showing you the process with just this one ROI. So you might have, when I described what our code does, you might have thought, well, white matter exists in white matter, gray matter exists in gray matter. How do you actually intersect the two? So how we solve this problem is that we dilate the ROI in a very specific way, and that we don't just inflate it, we actually project it into the white matter, so perpendicular to the surface, such that you maintain that anatomical specificity. So this green version that you see here is the same ROI, but just projected into the white matter. Now, using FreeSurfer information, we segment the gray matter white matter interface, which is that green outline now that you see surrounding the white matter. And then we intersected that dilated ROI with the gray matter white matter interface. And I don't know if you can see it in the back, but there's this little red outline right here, which is our gray matter white matter interface that is specific to that functional ROI. And you can see that it's tracking it very well. It's not really extending far beyond where you expect that boundary to be. Now, to find the streamlines that connect with that ROI, we do what's called a forward search. So at the endpoint of each streamline, it's going to look some small amount of millimeters, maybe up to 4. And if the streamline, when it's searching forward, intersects with the white matter interface, it will save that streamline. So just to show you what this looks like. So here we're looking at the inferior longitudinal fasciculus, which you can see in a better context on the top. So this red version that you see up here is the ILF that is intersected with the gray matter white matter interface of the ROI, which we expect to be more specific. And then what you see here in cyan below is when the streamlines are intersected with the original ROI. So it's that part in the gray matter. And what you can notice is that the cyan one has like a lot more stray streamlines, which is not good, and you're losing some specificity in that respect. And here it is again from a different view. So now you're looking at it from a bird's eye view, and you can even see a little more clear that there's like whole different part of the cyan bundle that seems to be included when we probably don't want it to. So this is like a little more specific. So now here's the video showing the track that we end up extracting in relation to the entire ILF. And you can see that it does kind of end up how we expect in that. So this is our FROI, this kind of blob right here. And you can see that we don't get any streamlines that extend behind it, ones that will go all the way back to the occipital lobe, because we don't want that. We only really want the streamlines that intersect here. And presumably most of these are going to go forward. So this intersection with the gray matter white matter interface is really important because it's not always done, and it's a little more conceptually grounded in that that's where oftentimes the streamlines are seeded from. So that's where you would want to check the intersection for. So far, and this is going to be subject to change as we continue developing the software, but it only requires a few dependencies, including MRTricks, which is a primarily diffusion software, and FreeSurfer. In terms of the data that you need some kind of functional ROI, diffusion MRI, and anatomical outputs that are in the same space. And presumably they should be in the subject space. And these FROIs can actually be defined on either the volume or the surface, but it's better to start was the surface, because you get that specificity that you don't get with typical voxel volumetric space. So all of our development is happening public on GitHub. Currently, even though it's not on PyPy yet, you can clone the repository and install it with pip. And we have a command line interface. You can see down here below just what it's like to actually call the function. And we do have a good set of feature plans, or our extensions. Right, okay. So we want to continue testing, building up our documentation, adding increased flexibility for different streamlined file types, do some parameter searching to make sure our default values make sense, adding an option to actually run tractography between two ROIs. Currently we have tractography as an input, but you can imagine some people might want to make the tracks between two ROIs. And then we'll eventually publish on PyPy and Docker Hub. And then we'll try to do some empirical studies and see, just really put it to the test. So in terms of what we've all learned on the team is how to set up a Python package using GitHub to version control and work collaboratively, and wrestling with some of our software dependencies such as Emmertrix, FreeSurfer, DiPy, and Fury. So thank you.
Thank you so much. The next team is the Connectomics Harmonics, which should really be a name of a band or something. What was your button? For the computer? Oh, no.
Thank you for your patience. We are Team Connectome Harmonics, and this is who we are. We are a team of 8 people. And just as a quick refresher, so I'm going to go through this really quickly because as we were practicing, we're running short on time. So let me know if I'm going a little bit too fast. First of all, this beautiful, trippy, psychedelic GIF actually came from the supplementary material from the paper. So it just kind of shows, at least to me, like how beautiful and interesting some of these concepts are when you really think about it and really visualize them. Just as a quick refresher, Connectome Harmonics is built off of this idea that vibrations vibrating through a physical body will produce these standing wave patterns at certain frequencies. This is sand on a metal plate with varying musical pitches being played through them. As you increase the frequency of the pitch, so you increase the complexity of the pattern. This is applied to the human connectome. And to the left, we have animal plates or metal plates that are shaped like animals, from which we can derive these very natural looking animal coat patterns. And in fact, a lot of nature uses these same patterns to self-organize systems into natural images like that. So this same technique is applied to the brain, and it shows how oscillatory activity throughout the brain creates resting state networks and by extension functional state networks. And this plot on the right is the first real meaningful plot that we came to in our hack week this week. And this was taken by taking adjacency matrices of DTI scans from people ages 18 to 88, because we were looking at the difference in connectome harmonics, how they vary by age. But this is from a single participant. And as you can see here, the first harmonic really looks very similar to the first harmonic on the original paper. And as the harmonics increase, you can see an increase in complexity. And the slides aren't working. So, okay, cool. And this is from the excuse me, the original paper as well. And you can see that you can predict resting state networks based off of combinations of these harmonics. And what we're looking at is how these harmonics change with age. And Brent will tell us a little bit about our data set.
Am I coming through? Okay, so we used the CAMCAN data set, which is the Cambridge Center for Aging and Neuroscience. It was a healthy sample of individuals between ages 18 and 88, at least for our specific sample. And they collected a large battery of tests, but primarily we were looking at the structural connectivity and their age to figure out what harmonics or what changes in the harmonics we might be able to observe. So the panel on the left is just the regular adjacency matrix. The nodes are from the Human Connectome Project parcellation. So there's a total of 376 nodes, the Glasser parcellation, as well as 16 subcortical structures that we added, or that were added. The middle panel shows the same subject, but sorted into the Yo resting state networks. So Yo 2011 defined 7 canonical resting states that have a pervasive name in the literature. That was similar to the previous figure showing the different harmonic contributions in the original paper to those networks. So we use that to kind of sort some of the outputs we'll get to in the next couple slides to see what they were. And yeah, so the individual harmonics you've already seen. So this is for the 376 regions we had, each column is a specific harmonic. So because we're interested in looking at this across age, we compared the computed harmonics and took the first harmonic from each individual and put them out side by side here. So you can see the top panel on the left, the bars indicate the decade range, and this is each subject's first harmonic. The second one here is much more interesting because you see the very clear left-right delineation between the cortex and the subcortical at the bottom for this kind of primary axis as far as this harmonic being the most associated with the left-right distinction. So moving on, we proceeded to sort them back into the resting state domains that I just described. So you can now see here, going across this plot, you can look at the harmonic basis by age within a specific network. So you can see in this first harmonic, the limbic subnetwork has kind of a lower contribution, seemingly. It took a little while to get here, or we would have binned them a little bit differently. And you can see as these higher order harmonics happen, you have some harmonics that do not matter for something like the visual network, whereas some where they might matter more for frontal-parietal, and that changes. You also get more noise as this progresses, as each subsequent harmonic is a weaker signal, so it is a more niche vibration within the network than the ones previous. So this was kind of our initial breakdown to just sort of try and visualize what changes we might be able to see in just the basic harmonic signals across age with the data that was most immediately available from the CAM-CAM. And this is just showing that as you get into higher and higher orders, they start getting noisier.
All right, and then we computed the eigenvalues for each of the harmonics. And as you can see here in the first harmonic here, we are seeing like for the 10th first harmonics, we're seeing like, and we're plotting eigenvalues and age. We're seeing this trend of decreased eigenvalue as you increase age. So we see that for the first 10 harmonics, and these eigenvalues, they might represent the complex of the connectivity in the brain in terms of structural connectivity. And here we plot entropy and age as well for the 10 first harmonics, and we see also that as you increase the harmonics, you increase complexity as well. But again, we see that there might be a relationship of age and entropy in the brain, and those also represent the connectivity in the brain. And we see that maybe when you get old, you also lose complexity in the brain. So our hypothesis was that when you're young, you have more connectivity in the brain, structural connectivity, and when you get older, those connectivities get less complex. So that's what we see here. And now is going to show some other graphs that we plotted.
All right, thank you, Arthur. And so to better visualize the point made by Arthur, we also computed the mean of the entropy grouped by age using a bin size of 10 years, as Brent mentioned, in the duration of 10 years between the data collection for the subjects. From that, you can see the lower harmonics numbers being uniformly lower than that of the higher harmonics numbers almost everywhere on the left. Then on the right, we subtract the mean of the left plot, which gets us the interesting insight that almost all of the harmonics entropy drop after the age of 58. Then the next slide shows that we also performed the same procedure on getting this visualization of mean of the eigenvalues grouped by age, which bears the same trend as you can see on the right, with the caveat that the trend seems to be more uniform than that of the mean subtracted entropy values as shown in the last slide. And then next, it will be the future directions, which Sai will be talking about.
Right. Thank you. And really briefly, some future directions that we would like to take this work. There's other analysis techniques that we didn't have time to try in this week. We did a little bit of clustering, but we didn't include it in this presentation. We'd like to do a little bit more principal component analysis and maybe write a paper about it. There's other data sets that we like to use. For example, the Human Brain Network contains diffusion tensor imaging information about children. So we would like to extend the graphs that we were showing backwards in time to see how children in development might, their harmonics might change over time too, because we have a couple of different intuitions on how that might work out, but we're not completely sure, and we would love to explore that. And also, the author of the original Connectome harmonics paper also did a paper about the psychedelic drug LSD and how it increases the higher order connectome frequencies and suppresses the lower ones during the psychedelic experience. So we're interested in other lifestyle choices that are like more long-term sustainable than that, that might be able to increase the higher order um, harmonics over age and possibly correlate those healthy lifestyle choices with preservation of those higher order harmonics, which, as we saw before, decrease in their entropy more than the lower order harmonics. And we already know a lot of lifestyle practices that can keep you sharp, older in age, but it would be really interesting to see how that affects the harmonics as well.
So I would like to acknowledge Alice that helped us so much with all these concepts, Noah and Ariel that helped us and make that possible. And I would like you to invite you guys, if you are interested in taking part of that team, we are thinking about like extending this project to future studies. So if you're interested, just come to Neuroharmonics, and thank you so much.
Great. We're gonna take a short break before the next talk, but before we go and do the break, just a couple of notes, technical things. This recent talk just reminded me to mention that if you're interested in continuing to work on the project that you're working on, there are a few things that you need to know. The one thing, important thing to need to know is that the hub will go down latest August 10th. So if you have stuff that's on the hub, get it into GitHub or get it somewhere else. Slack will continue to run, but it will go into the free mode sometime next week. And that means that messages older than 90 days will start disappearing. So as you're continuing your conversations there, just bear in mind that after 3 months, in 3 months, all the conversations we had this week and last week will just disappear into the ether of Slack. So again, if you have conversations that are important for you to store them somewhere else. That said, if there are resources that you need, anything, any way that we can support you continuing on your projects, please let us know. We're excited to have you continue to collaborate. Part of the goal of all this is for you to meet new collaborators that you'll continue to work with. You don't have to continue to work together, but if you do want and we can help you in some way, just let us know how we can do that, what you think, or if you have if you're not sure, just ask. Now let's take a 10-minute break and come back for the next round of talks here. So 10 minutes. Are you logged into Zoom already? Yep. So you can share a screen. Yeah, then you can start presenting. All right, and you can stand like this to present. So don't turn towards there. Instead, it's that way, so they can see, so the camera can see your face. Now you can be facing. I think you can't. I think it just stays there. Sorry, that's a Zoom. That's a Zoom feature. Okay, let me bring back it.
Can I be here?
Okay, we will resume the barrage of talks with NSD. One more NSD talk here. I don't think it's the last though. FS average.
Hello, everyone. My name is Anna. I'm from the University of Graz in Austria. And today I will be presenting our project called NSD FS Average, which is reliability of free surface reconstruction within subjects check. So about this project, we use natural scenes dataset from Allen et al. 2022. And this is a huge dataset which does not only contain high resolution fMRI data, but also a lot of structural scans, specifically for all 8 of the subjects. There were multiple T1 acquisitions acquired. We can see that most of the subjects have 6 T1 scans, one of them has 4, and one subject has 10. So having this data, we wanted to see how reliable free surface outputs are for each subject. And the idea was to compare surfaces within each subject. The problem with that is that each surface reconstruction within subject ends up in a different matrix size. So to solve that, we actually converted each subject surface reconstruction coordinates to FS average space, and then we converted them, interpolated them back to subject-specific space, because now we ended up with a standard FS average matrix size that we can then compare the coordinates of each run with each other. because we can track the index of each vertex. To compare them, we did Euclidean distance measurement, and then we did that pairwise for each run. Here we can see for each subject the kernel density estimation. And what can be seen here is that most of the run comparisons overlap. So the difference in the difference in the coordinates ended up being between 0 and 2 millimeter, and there are not many extremes. If you look at the these density estimations for each run separately, we can see that they look really good, really reliable. But if we look here, we see that at the cutoff of 5 millimeter, above that we have 0.027% of the vertices which have a difference in the distance over 5 millimeter. And that is actually more than 4,000 vertices, because one matrix contains more than 160,000 vertices. So we ended up actually looking for these extreme values. So we extracted the differences that are over 10 millimeters. We tracked those vertices, and then we created submeshes that we could generate on a white surface of the first run. And we saw some clustering, specifically happening in the temporal lobe and also in the frontal area. And if we look a bit closer, you can see that the orange submeshes are belonging to this white surface. So they belong to the first run, and the red submeshes are the ones that are differing. And specifically at the temporal pole, you can see how that difference over 10 millimeters actually looks like. And then if we look actually at the binary curvature maps for each run, we see, so here we have a lateral and medial view of 6 right hemispheres for the first subject. They look pretty similar, but of course, tiny differences are still noticeable. For example, here we see this protrusion for the first run, which we don't see for the rest of the 5 runs. Then maybe we can capture that better with continuous curvature maps. See, here we see the first run compared with 5 other runs. And again, we see that the curvature differences are the mostly pronounced in the temporal area and also at the frontal lobe. Then looking at the surface data, we looked at the gray matter masks and then compared the first one with all of the other runs for the first subject. And here in the middle of each plot, you can actually see what is the difference between these masks. And here, for example, we see again temporal pole difference, but we don't see a difference at the frontal area, which might be specific to the slice used here, but it can also relate to other things. And here are also some comparisons that we did for all other runs, and we see that these differences are really, really small. So we can here conclude that actually the first run was the one that was different, and what could be the causes of this surface reconstruction difference? It can be T1 noise, which can be caused by anatomical features of the subjects that are being scanned, but it can also be caused by acquisition procedure and things that happen in the scanner. It can also be caused by the reconstruction algorithm that FreeSurfer uses, because for each reconstruction, they start from a random point. So this randomization actually might be the thing that is causing these differences. All in all, we see that FreeSurfer is pretty consistent within multiple same subject MRI acquisitions, but still we saw some extreme clustering values. So the further analysis of other subjects would be needed to see how they fare. And yeah, so that was our project. I would like to say special thanks to Noah and Kendrick. So we used Noah's NeuroPython package, which made all of the interpolation extremely easy. And he also helped a lot with the analysis and plotting. And Kendrick basically designed the whole project and also helped out with making it happen. And if you want to follow the project, you can visit the GitHub repo. Thanks.
Okay, thank you so much. And I've changed the order. I apologize. I've changed the order just a little bit. So the next group to present is the experience sampling group, and that's just to accommodate some of our friends in other time zones. Okay, and you're presenting in Zoom, I guess.
Yep. Hey everyone, I'm on Zoom. Let me just make sure you guys can see this. Alrighty. I think you guys can see that okay. So hi everyone, Nico and I will be talking today about our experiences working with an experience sampling data set focused on unconstrained cognitive experiences and mind blanking during resting state. So we use two related data sets from Mordeheb Sapir, one on OpenNeuro and one on GitLab. The first data set we used included raw data reported in a 2017 paper in which researchers Van Calster and colleagues collected data on the neural correlates of unconstrained cognitive experiences. Van Calster and colleagues first developed a model of cognitive experiences based on a smaller sample of N of 18 to later use in a classification model. You'll see in this first image here that there are various experiences for various attentional states, but this data set primarily focuses on 4. So I have those symbolized with the arrows there. So perception, stimulus independent and stimulus dependent thoughts, as well as the absence of cognitive experience or mind blanking or mind wandering. And then in the second experiment, Van Calster and colleagues used fMRI to probe cognitive experiences and monitor resting state brain activity within the default mode network, dorsal attention network, and ventral attention network. In the second image here, you'll see the resting functional connectivity experimental paradigm that was utilized. And while the participant is at rest in the scanner, a probe, the exclamation point, was sent every 30 to 60 seconds, which prompted the participant to report on the cognitive experience they were processing at the time of the probe. And they also reported their attentional state as well. So in the second data set, we explored the cognitive experience of absence or mind blanking or mind wandering was the focus. The sample consisted of 36 subjects. This image shows a more detailed graphic of the data acquisition process and the statistical means through which data analysis would be performed. So connectivity matrices were estimated by means of Hilbert transform and phase coherence analysis using 2 pre-probe and 3 post-probe volumes for the analysis window. Recurrent brain patterns were determined with k-means clustering, and cosine distance would be used to determine similarity between report-related connectivity measures and brain patterns. That leads me to our exploration of this data set. So if you recall from the pitch earlier this week, I really had no clear idea whatsoever about what to do with this data. I've done GUI-based functional connectivity analysis for task-based fMRI, and I've worked with MVPA many years ago for an undergrad paper. But with that, I approached this data set with a very open mind and arguably too much ambition in certain areas, but I digress. Of these ideas, Nico and I decided to stick with 2. So 1, analyzing the data and trying to replicate Sapir's analysis pipeline. And then 2, applying these findings to a classification model to replicate Sapir's classification of mind blanking or mind wandering through functional connectivity data. So it leads on to the analysis pipeline. So I mentioned earlier that I've only ever run a functional connectivity analysis using a GUI. So I've used Column Toolbox. So doing this analysis computationally through Python was very new to me. I've listed all steps of the analysis pipeline here, but I'll be focusing on those that I spent the most time hacking. So the first step is the functional preprocessing stage, which I didn't replicate due to time constraints, but I used preprocessed data from the second data set I mentioned, so the mind blanking or mind wandering sample data set. From there, I tried to run global signal subtraction and regression calculations, but was unsuccessful in replicating due to a persisting error. I'm pretty sure this is because I just missed something along the lines in that pipeline process, and I think I know exactly what it is, but regardless, I moved on to the pattern generation and clustering code to see if I could replicate the connectivity patterns of mind blanking. I did already have process data to work with in this regard. So I was successful in doing that. So as you can see here, this image demonstrates the 4 recurrent connectivity patterns underlying mind blanking or mind wandering during resting state. This image is a direct replication of the matrices constructed by Sapere, even though there are no brain labels in this image, which is one of the major differences. What you're seeing here are all of the related voxels, well, significant voxels that mutually contribute to mind blanking and resting state. So while I didn't complete the pipeline entirely, going in very little information of how these analyses are represented computationally, since I'm so new to this, I learned so much about what happens behind the scenes in terms of functional connectivity analyses, which was very important to me. I only have one replicated image here, but this one image I feel well encapsulates all that I learned this week, working with this data set and working with Nico and better understanding the functional connectivity analysis pipeline. And on to Nico. If he's here. There you are.
Sorry, I'm here. So, well, following what Jennifer said, we were able to replicate the analysis of the functional activity analysis, and I was able to replicate the phase-based coherence matrix computation that the original paper did. And from that, I was able to, one of the goals of our analysis was to make classifications of mind-wandering states. So the original paper made classifications of mind blanking, so try to classify the mind blanking state, but for us it was much more interesting to classify mind wandering, that is a more relevant experience state using the literature. So we did that. Based on this replication, I convert the matrix into DID, thanks to the help of Ariel today that will help me work that out, make that up. So I apply a random forest classifier for that. This is actually the figure of the random forest classifier, and then we score it using the ROC curve. So next slide. So actually, and this was actually expected because the original paper for mind blanking state also got an accuracy super high. We got an accuracy of when we compare mind wandering. Oh, this says destruction, but this slide is against all other states, all other experience sampling states. The accuracy was almost 1, was 9.999, 0.999. So it was incredibly high, and it was cross-validated. This is just one run, but it was cross-validated, the one that is shown here, and the accuracy was always above 99. The original paper also got these values, 99, 98, 99. So this was sort of expected based on the previous study. We changed it to mind wandering, that we expected to get better results because we had more data for mind wandering than they had for mind blanking. And what we did here, and this was the most interesting part for me and what I learned to do, I used these SHAP values that show you the influence of each of the features on the output of your model, in a different way of what actually feature importance of the model is, based on sklearn feature importance. And it provides you these beautiful plots that show you which of the features impact on the impact of the model. So, and you can see based on the color that when that feature increases, so when it's more reddish, the impact of the model increase, or when if the values of the connectivity decreases, the output for mind wandering increases. So it's informative, and you can see that one of the clearest things that you can see based on the networks that appear, because if DA is dorsal attentional network, DA is ventral attention, DM is the full mode network, etc. So the networks that appear the most as the most significant, these are the 15 most significant features that we had. We have a list of more than 4,000 patterns for this connectivity analysis. And the 15 more important, most of them are between dorsal attention and the full mode network. That was sort of expected based on the literature. And we also computed the same analysis, but can you switch to the next slide? Yeah. And here in Ray, it is against destruction. Before it was against all other states. And again, the output of the model was super high. The classification accuracy was almost 1. And the SHAP values here, again, we can see that default mode network is the most prevalent. But here we can see that the cont, we also can see here that the cont network, that is the executive control network that before was not there, to appear, indicating that when you classify between mind wandering and stimulus dependent analysis, that is what is called destruction in some part of the literature, you can see that it changes what are the networks that are most relevant for classifying mind wandering in this case. And also you can see in which direction this is going. So this is something that I have never worked. I have never worked with functional connectivity. So all this for me was new. I had experience classifying mind experience sampling analysis, but with EEG. So this was part new, part novel. So I think it was interesting, and this is basically what we did.
Thanks, everyone. We're done.
Fantastic. Thanks so much. Next is the mentalizing and internalizing team.
Sorry, thank you. Hi, I'm Victoria, and I'll be going through the kind of like the intro for our project. So we kind of built off the pitch that I gave earlier this week, and we're looking at associations between resting state functional connectivity of the default mode network and internalizing symptoms in the adolescent brain and cognitive development study. So just really briefly, for the purposes of this presentation, mentalizing can be thought of as like kind of self or other processing or understanding, and internalizing is kind of a category of symptoms or depression, or symptoms or disorders related to depression and anxiety. So we see a lot of evidence from behavioral research that there's altered kind of mentalizing processes for individuals with internalizing symptoms and disorders. So we see kind of abnormal self and social processing. And we've actually seen that it's a prospective risk factor for the development of internalizing disorders, which tend to develop in adolescence. And we also see big differences in behavioral endorsements of mentalizing and internalizing disorders, in gender differences. So if you look at it from kind of a brain perspective, we also see differences in resting state functional connectivity within the default mode network that are associated with internalizing symptoms and difficulties. So in previous work, we've seen, for instance, hyperconnectivity of the default mode network to be associated with internalizing. Yeah, it's just sorry, bad slide. This is just existing research showing this. And this is just kind of like a basic visualization from Neurosynth, just showing that there is some similarity between kind of the mentalizing network and the default mode network, which is like helpful if you're trying to think about the conceptualization for this project. Okay, next. So as I kind of alluded to before, adolescence is really an important period where mentalizing strategies tend to develop, right? But it's also a period of really increased risk for internalizing symptoms and disorders. And we actually see that gender differences in, we see gender differences in mentalizing, and we see them in internalizing as well. So girls compared to boys endorse more hypermentalizing on average, and also have 2 to 3 times the risk of internalizing symptoms. So the Adolescent Brain and Cognitive Development Study, which we all kind of know about, presents a really nice opportunity to look into this. because they have both brain and behavioral data, clinical behavioral data across several time points following more than 10,000 adolescents. And so somewhat recently, we came across this paper where they sort of tried to look at this association at baseline in the ABCD study. So essentially, they found a really interesting finding, if you look in the upper right, where less within network default mode network connectivity was associated with greater internalizing symptoms, which is a little bit contrary to some of the previous research that I mentioned before. They also found a relatively low rate of internalizing symptoms in the sample. Only 12% of the sample endorsed one or more symptoms. So what we wanted to do was using not just baseline, but the longitudinal data that's available for us from release 4.0. We wanted to look at this kind of longitudinally. We wanted to include social anxiety in our conceptualization of internalizing. And we also thought that maybe part of why they got this kind of unexpected finding with the default mode network functional connectivity is because we need more granularity in kind of what we're measuring there. So we were hoping to get more specific features using the Distro Atlas as opposed to the one variable of like average correlation from the Gordon parcellation that ABCD provides. So we put together an 8-page pre-registration on OSF, which I really swore we weren't going to do, but I'm really sorry, team. And we had 3 main aims. So I can go next slide, and I'll just talk us through the first aim. So this first aim is a really, really simple extension of what the previous study found, but we're just using data from, internalizing data from time point 3, technically, but 2 years after the study, the Albertina study that I previously mentioned. So if you go to the next slide, you can see the model that we tested and what we found. So kind of similar to what they found with baseline variables, we found that reduced within network default mode network connectivity at rest was significantly associated with internalizing symptoms 2 years afterwards, after their scan. And we saw significant like main effects for sex and age, which are kind of expected based on sort of the literature that I described earlier.
Okay, so our next aim was to assess the association between within network DMN functional connectivity and internalizing symptoms using longitudinal data. So instead of predictive, just looking longitudinally. Here's the model we used. We are using random effects of site and relatives in order to account for stuff like people having twins in the study. Just to note, we ended up not being able to use those variables as well as motion as covariates or random effects because of some data availability issues, but we did end up including all of these things in the model for now, and we'll update it later. But basically, we also saw this inverse relationship between DMN connectivity holds longitudinally, which is essentially a replication of the study that Victoria was talking about, but longitudinal, as well as sex effects and an interaction between time and sex. Our next aim was to build machine learning classifiers to predict these internalizing symptoms from multiple indices of resting state functional connectivity. So how are we taking into account longitudinal data using machine learning? We wanted to classify the evolution of these subjects over time, since we do have brain data over time, based on their resting state functional connectivity. What we ended up using was called multi-label classification, which is a variant of classic classification where multiple binary labels are assigned to each instance. Here's a beautiful graphic of this that Alice on our team made, where basically you can see that you can exploit correlations among targets in order to create a chain of binary classifiers. So the predictions of each model are passed forward to subsequent models in order to be used as features in the next model. So you have these initial binary classifications, which are then passed forward. So in this example, Alice and Bob are being iteratively classified until you get a baseline prediction and a follow-up prediction as the classifier output. And I'm going to hand it off to Catherine.
Thank you.
So for the machine learning portion of the project, we used 2 different sets of features to predict internalizing. First, we used within and between resting state network connectivity as defined by the Gordon parcellation with 91 total features. And then given our specific focus on the default mode network, we used DMN specific features using the anatomical atlas with 24 total features. And our outcome, we ran the models both with just a simple binary internalizing outcome at one point, at one time point, but then also ran the models, and that's what you see is called multi-label, where we use both time points for internalizing. And so the models that we ran were basically logistic regression, random forest, a gradient boosting classifier, and a simple feed-forward neural network, both with the multi-label and the normal models. And these are the results that you can see here. So you see the model, and then we trained the models on the training set and calculated the accuracy on the test set. And just a quick note, we did run the logistic regression with all two-way interactions between all of the variables with lasso regularization, but that took too long to run, so the results aren't included here. And these are all considered preliminary as well, because we didn't get to include all of the confounds that we had wanted to, but they did run, and we saw that we got pretty high accuracy. So we were really happy with this. A couple things to note is that we got really high accuracy with just the plain logistic regression. So if we run the final models and the simpler model is the same accuracy as the more complex one, we would choose the simpler one. And then the multi-label models are lower accuracy just because it's taking into account those 2 time points. And so it's a more complex outcome. And so that is what we expected for it to be a little bit lower. And then this was an important part for us and will be an important part because we're not just interested in creating classifiers with the highest possible accuracy, but we also want to understand which features of these resting state networks are relating to internalizing. And so these are just a couple examples of the things we looked at and plan to look at. So for example, in the random forest, we can choose the top predictor, which is how many times each variable is used as the root node. You can see the results here with complicated variable names. So we're not at the stage of wanting to interpret yet, but that will give us an idea of the most important predictors. And then we'll want to explore, like the previous team said, the SHAP method and other ways to extract the top predictors. And there's still a lot of work to be done. Obviously, there's a lot to flesh out with hyperparameter tuning and accounting for confounding variables. And we used binary classification, but we'd also like to try out regression models using internalizing as a continuous graph.
So after these 3 initial aims, we were also planning, in our 8-page pre-registration, we also put a 4th aim where we were planning on looking at functional connectivity features that we have studied with the machine learning models that Catherine introduced in as a cohesive singular graph structure. So as you can see in the figure here, the idea was for each functional connectivity to take into consideration the weight of different connections between the ROIs and treat each graph as a single vector point for a machine learning classifier. There are some methods that are out there that have been invented in the last couple of years, but looking into the literature and talking with Noah, we found out that there are actually no well-established libraries out there yet that deal with fully connected graphs that are also weighted, because a lot of these methods take into consideration the structure and not the weight that is given to the edges. So we might think of doing this in the future.
Yeah, so I'm going to give a brief summary of our results. And so the results of the preliminary analysis show that there's like a negative relationship between the deformal network connectivity and the internalizing symptoms and they are in the different time point. And we also use a longitudinal modeling to confirm that the Albertina's results, they are also held. And we also found a sex main effect and the interaction effect between the sex and time point. And we also use multi-label machine learning approach to find out, oh, the results turn out to have a high accuracy from the classifiers. And so the next patch will talk about the future directions we may thinking about to focusing on like the subnetwork or to focus on the specific symptoms to see if there's any unique effect from those subcomponents and use a lot more rigorous experimental design, like control for more variables and look for another method to improve our way. So I think that's enough for our presentations, and thank you guys for listening here.
Okay, that was great. Now we come to the IEG section of the presentations. I think the first is the IEG bipolar referencing. I hope everyone has a slide with references with that such a small font for everyone.
Okay. Can you hear me all right? Sounds like it. Cool, sweet. Hi, everybody. My name's Val. If I look like a stranger, it's because I was on Zoom for the first week and a half of all this. Finally made it in person. This is the rest of our group here. We did IEG bipolar referencing for our project. So if in the last couple of weeks you're still unfamiliar with what IEG is, it's electrophysiological signal that's obtained from implanted subdural or deep electrodes. If that was not a mystery to you, but bipolar referencing is a mystery to you, that is simply computing the difference between neighboring electrodes. Even more mystifying perhaps is what the problem actually is. And it's for us, we wanted to know, can we automatically determine which electrodes are neighboring?
So this might sound like a simple problem, but I'm going to explain all the ways that it is in fact challenging. So we're provided with, from the BIDS format for EEG, we have a list of channel names, and then we also have their XYZ coordinates. So we can end up plotting something that looks like this on the brain, seeing all of these different electrodes. But one of the first challenges is that we could end up with these overlapping grids. So for instance, we have the grid that's highlighted in light blue and the grid that's highlighted in dark blue. And we can see that there's electrodes that are very close to each other, but they shouldn't be counted as close to each other because they're actually from two separate devices. So that was our first problem to try to solve. And then the second is that we only want to consider electrodes that are directly next to each other in either a horizontal direction, so like these two next to each other, or sorry, in a vertical direction or in a horizontal direction. So there's kind of two main challenges that we have to kind of solve. And so the way that we did that is we started out by computing the 3D Euclidean distance between all of our electrode pairs. Then using the channel names, we're able to actually separate the arrays into their different device names. We found a threshold for what the distance between the neighboring pairs should be. And then we're able to match the pairs based on those distances. And then finally, we did some beautiful visualizations. I'm going to hand it over so you can see what those look like.
All right, so just to, again, to reiterate, what we were able to do from the 3D coordinates was plot where each electrode sort of sits on the brain. And then we were able to use with histogram, after we calculated Euclidean distances, we were able to compute the thresholds that were necessary to sort of cut off the distances we did not want to include, but the pairs that we did. And then we're able to generate sort of this like directionality plots in which we can say, okay, these are the neighboring ones to consider if we're looking in the vertical direction, or in the opposite direction, these would be the neighboring. And so we were able to calculate these for the pads or the devices that are in sort of a grid shape. And then we just generate this in a nice plot over here. And then also we're able to do the same thing, next slide, please, for plots that are stereo electrodes, so that they only have the one directionality. So yeah, that's what we were able to plot. And these are some nice visualizations. Again, just again to reiterate, it's showing the directionality in which you need to consider their bipole connection. And then, yeah, that's that.
Nice. So one key thing that we were hoping to be able to do, and are maybe kind of close to being able to do, is contributing this upstream to Python MNE. So as a part of that, we wanted to make sure that this was as reusable, automatable, and modifiable as possible. So the first thing was making sure that we were compliant with the IEG BIDS standard that's still sort of in work in terms of the data that we get from OpenNeuro, and also just the nomenclature and what things are called, so that when someone looks at our code, they can know what we did. So we actually spent a decent amount of time taking the existing sort of prototype code and refactoring it so that the kind of naming and variables made sense and it was well documented. And the nice thing about that is then we also know the upstream existing MNE method that already exists. So we kind of made sure that our outputs are in the right format, so that can be subsumed. So ideally, this should just be like something in the background. And yeah, we recently moved from sending code snippets to each other on Slack to GitHub. So we're pretty proud about that. And so in terms of next steps, we basically did this all prototyping with one subject from one data set. So we want to expand to multiple data sets, of course, like I think a lot of people here. Also, the kind of documentation is an ongoing thing just to make sure that everything looks good. Also, other like software engineering stuff, automated tests, making the code more modular, that type of thing as well. And then theoretically working with whoever the owner of Python MNE is to understand what the gaps are between what we have and what they would like. That is that. Thank you.
Very cool. And now the IEG fMRI connectivity team. Are you connected to Zoom? Beautiful. Great, here you go.
Hi, we are from IEG fMRI Connectivity Group. We are a group of 6 people from very different backgrounds. Yes, the data set we used was the OpenNeuro data set that was published this year, where they used both IEG and a functional MRI in the same subject performing the same task, which was watching a short movie. So because this data set contained both IEG data and fMRI data, we thought we could maybe look at those similarity and dissimilarities between two modalities. Yes, so that was the main objective of our project. We decided to look at the functional connectivity between two modalities because fMRI data only contained movie watching period, no resting period. So we only look at the task period. We first selected 10 subjects that have both, of course, fMRI and IEG, who are right-handed, and electrode coverage was on the left hemisphere. And we only selected ECOG electrodes, and we didn't use like depth electrodes or SEEG electrodes. So to reiterate, the main objective of our project was to investigate the similarity in functional connectivity measured by IEG and fMRI. Yeah, this is the overlook of our method. So because because the fMRI was done on whole brain and our echo electrodes were all different across subjects, so we first had to co-register ROIs using electrode coordinates, and all the data were raw data, so we had to do all the pre-processing and then look at connectivity in IEG and both signals separately and try to compare these two matrices. So for IEG pre-processing, we first dropped bad electrodes marked by the researchers and only used echo electrodes, and then we performed Harman average referencing and to remove overall noise. And we extracted different frequency bands for broadband or just over 0.1 hertz, and theta band 4 to 8 hertz, and alpha and high gamma. And this is the example subject echo coverage on the surface.
Cool. Yeah, so every subject that we used had both ECOG data and fMRI data. And yeah, neither were processed. And so something we ended up spending a little bit of time on was doing the fMRI processing or pre-processing for all the 10 subjects, which involves surface reconstruction. We used fMRI prep on a singularity image. This is actually good, I think, at least in my lab. I tend to use data that's been processed, and often a lot of the open data. This is like part of using open data, I think, for us, was that you don't have to actually implement all this stuff. And so anyway, we did pretty normal stuff, but we had to actually like make it and stuff. So for me, that was a good learning experience. So yeah, we did all the standard fMRI prep defaults, and then did some processing specific to functional connectivity, regressed out nuisance variables, the low pass filter and demean and detrended the data. And we kept all just as we just mentioned, we kept all the subjects in their native space. So we're looking at their like vertices on their specific surface, which we're then going to map onto the electrodes, which Graham's going to tell you about.
Okay, so we were given we wanted to look at the connectivity using a one-to-one comparison of the electrode coordinates, so where we're recording all that electrical activity, versus bold signal in the exact same locations. We were given the electrode coordinates, and those were actually registered to their MRI space. But the problem with that is that they were hovering a little bit above the brain. And when you're doing fMRI connectivity, you need those regions of interest to be on the brain. And because we were doing a surface-based functional connectivity analysis, we wanted those to be on the surface. And so what we wanted to end up with was something that looked like this. So each of these different colored regions are regions of interest that we can run these functional connectivities to. So we can extract the average bold signal in these regions and find the correlation between all the regions to make a correlation matrix. So the way we did that was first we took these coordinates, then through a little bit of math, we were able to find what the closest vertex on each individual brain was. That's the vertex from the surface that we created with FreeSurfer. And we created these 5 millimeter radius little circles. And you can see on the inflated brain, those are circles. And then once you deflate that to look at the actual peel surface, they turn into these interesting shapes, right? So we did this for all the different subjects, and you can see that the different electrode placements were different for each individual. And that's one of the reasons why we decided to make this to do the analysis in the individual space, in the native space.
So we performed functional connectivity using pairwise correlation coefficient. And you get metrics, but because electrodes are all different from each subject and they are ordered in kind of arbitrary way, so we first had to find the right order by clustering these correlation metrics. We use a hierarchical clustering algorithm called Dendrogram implemented in SciPy. And for this is an example metrics. You can see that so before we ordered this metrics, there was no clear structure. After we reorder these metrics, we can now see clear structure in this one area. So now we use this order to order both IEG matrix correlation matrices and bold matrices. So for example, here we first did clustering, perform clustering algorithm based on high gamma. Sorted all the other matrices based on this metrics. Yes, you can see within across within IEG, these four matrices, there are some similarities, but with fMRI, the first column, we don't really see clear similarity. And yeah, this is on other, oops, sorry, the other subjects. And we could also order them by not only by high gamma, but also by other oscillations such as, oops, this should be, so the first one should be, so sorted, this one is sorted by high gamma, the other ones are sorted by other broadband signals or alpha or theta. And we get, so we compare these matrices using Pearson correlation, and we get negative correlation value, but we didn't perform a statistical analysis yet. So yeah, this is basically it.
Cool. Oh, wait, sorry, one more slide or two more slide here. Yeah, okay. So basically that's the results of the project. Okay. Is that you can look across these things, see like, okay, fMRI correlations don't really look so much like the correlations, even though we're in the same space. Okay. So we have all of our stuff on GitHub, and do we believe the results are right? I don't know, because we didn't really test that much stuff. So I think like if you get a good result or a bad result, you got to go back and test all your stuff and make sure things look good. So I think if we're going to keep continuing on with this stuff, we would go through and make sure things like look good. Like for example, you could take the little clusters we make, project them onto the brain to see like, are the things that have high correlation actually in places that should be correlated with each other, stuff like that. Do like a lot of the fMRI like we did some basic pre-processing, but there's a lot of movement in these subjects. We didn't do some cleaning of relating like seizure activity, and then we looked at connectivity and not activation. So there's a lot of stuff you could do here. And basically how I felt is that a lot of us came in wanting to use open data and wanting to learn how to do IEG. And so in that way, this was like very successful in that we were able to all work together with people that we just met using like a shared Amazon S3 bucket with different skills, and get to use IEG and fMRI and get them to work together. So that's our group. Oh, wait. Thank you. Thank you to everyone here, especially Ariel for help with S3.
Thanks a lot. And we will now take another short break. It's another 10-minute break here, and then come back for the last round of presentations. All right. Are you all presenting now?
Yes, I believe so.
Okay, I know it's hard because there's a big screen right over here, but it's the same screen over here. So it looks like this. All right, let's be seated, please, and we will continue here. You thought you were done with the NSD data set, but no, it's still here. All right, and now to more NSD stuff.
Hey everyone, so our presentation is called Task Performance and Individual Differences across Resting State and Natural Scene Memory Tasks. Okay. Okay, so we already talked a little bit about this data set today, so I'll just go ahead and go through it quickly, but we focused mainly on the continuous recognition task where participants went through 40 sessions, and through those 40 sessions saw between 9,000 and 10,000 natural scene like images, which you can see on the left there. And so for our project then, we use this to analyze the data set in context of memory using behavioral multivariate machine learning and connectivity tools. And just a little bit about what the data looks like. So you can see the 40 sessions there, and across them, we have the continuous recognition task, which was acquired, as well as resting state data. And for the behavioral data, there were certain features that were recorded, such as response time across the trials. And for the imaging data, we got resting state and also task-based imaging data.
So the first thing we did is look into some behavioral analyses, and because our project was very much motivated by the study of memory, we actually wanted to take a look at how well humans classify these images as old or new. So this is a term that we're probably familiar with from the machine learning literature. So you can see here that during this continuous recognition task, participants were tasked with saying whether or not they had seen an image before, and their responses can fall into these 4 response categories. So we also looked at reaction time, and we had a couple plots of reaction time that we made, but we didn't get into doing any neural analyses with this data. So for that reason, we'll just move past that. But looking at the kind of group level, we just took a count of how many responses each participant had that fell into each of these 4 categories. So you can see there are very few trials where participants failed to respond, and most participants had many more hit responses than others. But note that the orange and green bars are both correct answers in this. So really, to look at how well participants performed, we wanted to plot these along a curve with the true positive rate, so the number of hits over the number of times when the item shown was in fact an old item, plotted over the number of times they responded that it was old when it was not, over all of the times that the item was not old. What you see here is the 8 points on the plot are individual subjects. The orange diagonal is the chance line, and then we actually took a machine learning classifier and applied features of subject and whether or not the item was actually old or new. And using that, you can see that we get some classification. We didn't do any quantitative analysis on the classifier's performance or area under the curve, but you can see it's kind of approaching out where the actual subject performance is, even with just those 2 features.
So for our fMRI analysis, we focused on resting state functional connectivity. We did a few types of analyses. So we did seed-based connectivity, ROI parcellation connectivity, and whole brain connectivity. So for our seed-based whole brain connectivity, we created a mask of the retrosplenial seed. And we were particularly interested in this area because of its function with the memory gateway between the default mode network and medial temporal lobe in spatial and episodic memory, as well as navigation. It also has anatomical connectivity to the hippocampus, entorhinal cortex, the anterior thalamus, as well as visuospatial and other sensory cortical areas. So this is a single subject map of a 1 sample t-test. We have our seed in the green, and as expected, we found positive connectivity in the posterior regions like the precuneus, as well as some medial regions. And again, this is all within the default mode network.
So the next thing we're interested in is continue looking at a resting state connectivity, but looking into the ROIs and then looking into like what are some of the regions that we're interested in, like are they how strongly connected are they, or like connectivity strength are they? So we use the subject space, and then we found there's like a manually segmented medial temporal lobe that is already coming along with the data, and then there's also functionally localized areas like body, like in the vision area. So there's body localizers, face, and words, and they're just like briefly shown there for reference. And then we conducted this ROI connectivity analyses. So on the upper left, the bigger square is showing all of the medial temporal lobe subregions, and then on the lower right are the like the vision areas. So I just want to quickly point out that like the vision areas are kind of like the voxels might overlap with each other, so it might not be the like most optimum, but anyways, we're just want to look into this data set. And then so some of the question we can ask with this is that is the resting state connectivity consistent within subject before and after the task? And then does the subject network consistency change over time? So remember that like there are 10 sessions where they collected resting state, and they collect the resting state one scan like before participants participate in the continuous recognition task, and then another scan, another resting state scan after the recognition task. So we have like overall like 10 session of the before and after. So with that, we continue to do another correlation analysis of the top, so like the upper corner of the before matrix and the after matrix, to conduct like subject-wise correlation score. So just to reflect the consistency of these two matrix over time, or like over sessions. And then these are the plots or like the consistency for each of the participants. So I guess this is in a way that we can tell the connectivity, these areas connectivity consistency over time for all the participants. And along the similar line, and Zhenqi actually made this plot, is that we're also looking to the similar idea of whole brain connectivity before and after the task. And then what you're seeing here, like with the red squares shown is like each participant. And then we're seeing that there's more like intrinsic within subject correlation than between subject correlation.
Okay, and then we also looked at functional MRI data during the memory task and when participants were seeing the different images. And what we did for that is we used a classifier to see whether we could decode from the brain activity in the medial temporal lobe regions, whether an image was old or new. And that's what you can see in blue on the graph for these 10 different ROIs. And so you can see that decoding accuracy is above chance, which is the dashed line. And then we also looked whether we could decode whether a subject thinks the image is old versus new, irrespective of whether it's actually old or new. And these are the orange bars. And so you can see that they are generally higher and that we can better decode whether what the subject thinks, and not whether the image was actually old or new. And these results are sort of in line with another four-way classification that we did, where we decoded whether a given trial was a hit, miss, false alarm, or correct rejection. And you can see that we can also decode that above chance accuracy. And what is interesting here is to look at the mistakes that the classifier is making. So for example, this line here are all the correct rejection trials. And then we can see that the classifier gets it mostly correct. But if it makes a mistake, it most often says that the trial was a missed trial. So from this matrix, you can see that misses and correct rejections are often confused, and hits and false alarms are often confused. So again, it's more important what the subject thinks, and less important whether the image is actually old or new. And then another group member also looked at convolution neural network for decoding, and that's still work in progress. She got it to run for 1 ROI and 1 subject. I guess the intermediate conclusion is that it's sort of difficult because this data set is very unbalanced. You might remember from the beginning that there are many more hit trials than false alarms, and that's what makes classification quite tricky for this data set. Yeah, that's sort of it. And I guess we also learned a lot during this hacking week. And yeah, here again, all the team members, and thanks to the people listed here and for you for listening.
Thank you so much. Next up is the amygdala and pain team. On Zoom. Yeah, put it there.
Sorry, deciding where to stand. It's important. I'm going to talk about some work that my group did, which was looking at kind of risk factors for developing chronic pain, particularly with relation to the amygdala. Okay, so vulnerability to chronic pain. So quite a lot of people at some point in their life will get injured. You might kind of trip over some stairs. You might kind of attend your hackademy and hack for a week, and then your back is killing you. And then you kind of go through this phase of acute pain. And the thing is, a lot of people get better. They kind of get on to doing their natural life again, playing tennis. But like a little subset of people actually end up having chronic pain. So what is it that makes these people different? There's kind of a lot of different factors around. I would refer to this as being kind of that $1 million question, because if we could tell what made people resilient, we might be able to develop therapies for those who aren't. Although my PI informed me that was probably only about $100,000 question in a grant. So one of the different things for the risk of chronic pain are things like just generally your hardware at birth. What's your gender is. Women tend to be more vulnerable to chronic pain. There's multiple genes that might have an influence. There's also environmental influences. If you've had pain before, especially maybe when you were younger, if you've had lots of stressful and traumatic life events. And there's also kind of gene and environment interactions, including things like your personality and psychology. People who tend to catastrophize about pain are more likely to have chronic pain. And if you kind of like dig down into the physiology of this, you kind of have your innate mechanisms, acquired things, kind of like priming. And you also have these brain vulnerable networks, which is the thing that we're really going to focus on today. So why the amygdala? Everyone kind of thinks of the amygdala as being mainly just involved in like fear and stress. But, you know, chronic pain is kind of stressful. It's
not only a physiological stressor on you and your body, that's, kind of why you end up feeling so tired sometimes when you have pain. But it's also kind of things like it causes anxiety, it can cause social problems, and it can cause medical problems as well, even just things like your kind of massive copay that you might get from the hospital here. But it's kind of interesting as well in chronic pain in that it might be causing this vulnerability to get it. So there's this really cool study, which is here. Oh, yeah, you can. So it's here. So what they had was they had 3 groups of people, and what was really cool is that they followed them over 56 weeks. So they had the white circles, the healthy people. The black squares are people who had an acute pain that then went on to be persistent. And then the ones with the gray triangles are people who had acute pain but recovered. So what's really interesting here is that the amygdala volume for both the healthy participants and the people who had an acute pain but recovered is kind of the same. But for those who had ended up getting persistent pain, they actually had a lower amygdala volume at the start, and it seemed like it only got worse. So this might be kind of predisposing them to even more pain, and it might actually be something that's kind of an underlying cause. But this study only really looked at the volume of the amygdala as a whole, so both sides of it. But what's kind of really interesting, coming from the rodent literature, is that the 2 different amygdalas may be doing something really different. So things with the right amygdala, especially the central nucleus of the amygdala, seems to be pro-nociceptive. So that means kind of actually causing the chronic pain. And so higher activity in the central nucleus of the amygdala is associated with these kind of persistent pain states. However, recently there's been kind of some exciting findings. Like most papers were just thinking the left amygdala was like hanging around doing nothing. But actually it seems like it's been trying to work really hard against the right amygdala. So it seems that actually the left CEA might actually be reducing chronic pain. So in this kind of hypothesis here, they suggested that with acute pain, you get this, like the pink here is an increase in right amygdala activity, which slowly goes down over time. However, in comparison, the green activity is going up, which is the left amygdala, so it's protecting you. And then they're not really sure what's going on in chronic pain. Is it that the left amygdala is just not, the right amygdala is staying active and the left amygdala can't catch up? Or is it just that the left amygdala is being a bit lazy? So this has not really been explored in the human population very much, apart from this one study that I could find. So what they looked at was the volume of the left and right amygdala. So they had the controls, which were people with no pain, and the left is the blue bar graph. So as you can see, and I can't see, okay, I got it. As you can see, the left amygdala seems to be a little bigger, and the right amygdala is smaller. And as a whole, the amygdala volume seems to be lower in these people who have chronic low back pain. What was kind of really cool in that study was that the left amygdala difference between the controls and the people with chronic low back pain was far greater than the change in the right amygdala, suggesting that there might be a decreased activation in the left amygdala causing this chronic pain phenotype. But they only had 33 participants per group, which probably isn't a high enough power. So what we wanted to do was to look at how amygdala volume interacts with other vulnerability factors in chronic pain, particularly acute pain, because people often will have an acute pain that will lead on to a chronic pain, even if that was kind of an earlier point in their life. So we chose to use the Human Connectome project. And they had two different types of pain analysis. So both of these were acute pain analyses. So pain intensity over the past 7 days. How was it? Well, so kind of a sort of 1 to 5 scale, 5 being bad. And then also your interference, how much did it affect your enjoyment of life? And then we looked at the volume of the amygdala in relation to this. So this was our first finding. So we kind of wanted to just initially look a little like that first paper that I showed you. What was interesting for us is that we found that the right amygdala was actually tended to be bigger. And these are people who over the past 7 days had reported either like no pain, moderate pain, or high pain. And the left amygdala in all of them was kind of smaller than the right amygdala, but no different between them. However, what was interesting was when we looked at the difference in size of the left between people who don't have pain and people that do. For the left, you actually see this more significant decrease in the left amygdala volume. And that's also kind of seems to be the case when you look at high pain versus moderate pain. So this might be incremental. The next thing that we were interested in was age. People tend to get more chronic pain problems as they get older. And what's really kind of cool that we pulled out from here was that even though in the Human Connectome Project, people are quite young, only kind of really going up to about 36, 40, there was this really significant decrease in amygdala volume as a whole from your 20s into your 30s. And this is kind of a bit of a complicated one to see. But so what we were looking at was different levels of pain at different ages and if there was any correlation. So this is the kind of the 22 to 25 group. This one is the moderate pain of 22 to 25, and this is high pain, 22 to 25. Sorry, a bit of it got cut off. What's really interesting is that here you're seeing the volume differences between the right and the left, so right being bigger than the left. And it seems to be that there's a more prominent effect of pain when they're younger. So we could say that this kind of pain in people who are 22 to 25 is giving them the brain of a 36-year-old and possibly the same kind of vulnerability to chronic pain. So to conclude, we say acute pain is associated with greater differences between right and left amygdala volumes. Decreased left amygdala compared to right amygdala volume may lead to vulnerability to chronic pain, as left amygdala is thought to be protective. Amygdala volume decreases with age, and this may contribute to the greater vulnerability with chronic pain in the older population. It would be really cool to see it in a kind of older population than just 36. I think a few people would get offended if I said 36 was old. So then acute pain early in life might actually be aging your brain, and that actually might be causing your kind of predisposition to chronic pain. So I want to thank the team. Jen will kill me because she said to make her picture small, but she deserves lots of recognition. So Elam and Jen both did a lot of work on this project, and they're both on Zoom. And yeah, that's it.
That was great. Thank you so much. Now I know my ABCD is next. As we're presenting, look at this. Zoom stuff.
Sorry, just for the Twitter. Yeah. Thanks, Victoria. Okay, hi everyone. We are really excited to present to you our NeuroHack project called Now I Know My ABCD. And so just for some quick introductions, my name is Sana and I am a rising second year PhD student working with Dr. Deanna Green at UCSD.
Hi, I'm Claire. I'll be starting my PhD program at UCLA with Dr. Jen Silvers in a month.
Hi, my name is Monica. I just finished my PhD at Columbia with Dr. Kevin Oxner, and I'm going to be starting a postdoc with Phil Kriegel at Emory in 2 weeks.
Hi, I'm Lucy. I'm a second year at the University of Oregon working with Kate Mills.
Okay, so this is just an outline for our presentation today. So we're going to start with introducing a bit of our motivation and inspiration behind our project. Then we'll go into some specific aims we have. We'll walk you through the implementation details of our project, and then we will end with how we plan on continuing our future collaborations. Okay, so to put it into context, as you can see, working with publicly large data sets can be difficult. Many people have attended workshops and other training opportunities that discuss resources for how to navigate these types of projects. However, we have noticed that unfortunately, these resources are not located in a space that is easily accessible. And therefore, given the inconsistencies surrounding this problem, we have decided to initiate a resource hub for researchers working specifically with the ABCD dataset. So for those of you who do not know, the ABCD dataset is the largest long-term study on brain development and child health in the United States. And so this NIH-funded project has funded leading researchers in the field of adolescent development and neuroscience, and this is across 21 research sites. And so their protocol is a comprehensive set of assessments, which ranges from physical, cognitive, social, emotional, environmental, behavioral assessments, as well as multimodal imaging and biospecimen collection. And so that being said, this is an extremely rich data set, but with so many different measures and waves, it can be intimidating to start with. So our project has a couple of specific aims. So for aim 1, we hope and have curated a resource hub tool accessible for ABCD researchers. Aim 2 is to manage code troubleshooting and inform proper data sharing, and this is done through our GitHub discussion board. And lastly, we hope to inspire open science practices. So our implementation exists through multiple streams. So we have a GitHub repo called Now I Know My ABCD. We have a docs static website through Jupyter Book, also Now I Know My ABCD. And we have a Twitter pipeline, hashtag Now I Know My ABCD. And yes, we realize Now I Know My ABCD is annotated 3 times on this slide. We like the consistency. And now Lucy is going to talk a bit about the design.
Cool. So one of the first things we did was essentially to make this GitHub discussion board. So within our GitHub repository, we created this GitHub discussions page, which is essentially intended to help researchers find information about how to work with ABCD as well as to connect to other researchers. To make this discussion page easier to use, we have this 2-part organizational system. So you can see on the left, when users make a post on this discussions page, they can choose a category to put it in. And we wanted this to kind of feel like a combination of like Slack and Stack Exchange. So some channels are just for open discussion, people sharing thoughts. And in other channels, there's basically a Q&A enabled. So people can post a question and then actually like vote and select an answer that they think is correct. And on the right, you can see that we can also tag posts with different labels. I don't know if they're super visible, but there's like different topics, things like the like programming language that you might be using or like the topic of your study. And this basically enables researchers to search through all of these different discussion posts, either by selecting like all posts tagged about mental health across all channels, or going as specific as looking only like for code and stats help tagged with fMRI and like ABCD release 4.0.
Okay, so last year, myself and a few of the attendees here attended the Modeling Developmental Change ABCD workshop, and that's a 5-week intensive workshop aimed at learning how to produce rigorous and reproducible analyses within the ABCD dataset. And so, of course, we had a Slack workspace that housed channels related to code help and just other resources. Given Slack's new 90-day policy of deleting messages, this is problematic, and we would hate to lose all of the information in that workspace. So therefore, we used last year's Slack workspace as a pilot to jumpstart our GitHub discussion. And so we were able to export all the information from every public channel, consolidate and transcribe discussion threads into the categories that Lucy just talked about, and that's in our GitHub discussion. So as you can see, we were able to salvage a year's work of messages and information and find it a new home. So next, let me walk you through an example of what that discussion thread looks like. So this is our GitHub repo. So you go to docs, and then there's a tab up here that's discussions. So you can see that this is pinned. These are our guidelines. So it's a nice user guidelines. Read that first. On the left is the categories that Lucy just went through. And then these are the discussions that were exported from our Slack. And so we're just gonna take a really good look at this. Yeah. And now we're gonna we're gonna scroll, and I'm gonna click into one of these to show you guys an example. So we have brain RI visualizations. And so the question is, what is the best way to visually represent correlations between brain RI findings and clinical correlates when you want to highlight a couple of key significant variables? And so we have 2 answers, one in R and one in Python. And you can see that they're tagged with R, Python, and imaging, and they're categorized in the stats and code help. So this is a great example of what you can find in our discussion section. And here's Monica to talk about our website.
So our hope is that once some of these questions have been answered, the original poster has agreed this is something that worked for me. It's something that I would like to be written up so that people in posterity will be able to follow the code example. They can refer to the static website, which, as you can see, is rendered through JupyterBook, the beautiful package that we learned about through REL. And the book also auto renders on GitHub Actions and serves on GitHub pages. So what this means for us specifically is that even though I'm the only one with JupyterBook installed on my computer, anyone else on the team could push to the repo. And even though they don't have JupyterBook installed, GitHub Actions will render the website for all of us, which is super slick. We also have some brackets worked code tutorial in the book right now. For example, using the tips called from the Slack group about brain ORI visualizations, I've created a worked example with code that, don't ask me how it works, it's really ugly under the hood, does actually run code, inject code into this help page, and shows plotting outputs for both R and Python. I learned lots of Python for this. And so you can see an example as well of a exploratory scatter plot matrix that you might use before you know which correlation you want to focus on. And then when you know which one you want, you might choose to show it on a static brain atlas. One thing I do want to flag is that while I do very much love GitHub Actions, only the Python code actually runs through GitHub Actions right now. The R code, I still have to click the button, but that is a future direction.
Okay, so when we were thinking about this, we obviously couldn't leave out Twitter because academic Twitter, research Twitter is definitely a thing. Okay, so to do this, I created a developer account and was able to generate API access tokens. And I played around with 2 different Python packages. One is one is SNScrape, and then one is TweePy or Tweepy up to interpretation. They both function pretty similarly. TweePy was only able to go past the recent week and pull everything by keyword or hashtag, whatever. And then Essence Scrape went all the way back, but the output was a little bit messier. But I wanted everything ABCD, so I used Essence Scrape. Thank you. So I ended up doing like a preliminary text analysis and generated everything into subcategories. So they're listed there. And I generated this spreadsheet. And so you can like, it's all interactive. And if you see a question that you're like, oh, I also have that question, you can go and click on the actual tweet and read through the thread and interact with it there. Yeah. We all know what Twitter looks like. Good. And then I like was able to embed it in the website with the code right next to it. Okay. And then for more recent and active tweets, I generated RSS feeds, but they live behind a paywall. So we only are able to have one right now, but hopefully in the future it's also a great application. We'll hopefully be able to generate more with the actual hashtag now I know my ABCD. And there's Victoria's tweet that I was able to actually find from our website and click into and interact with. So yeah. Okay, so as far as, this is all to say that this is definitely a work in progress. We're really excited about it though. And as creators, we plan to like manage and monitor the functionality of it and also the guidelines. And we plan to have monthly maintenance check-ins. And then, yeah, we're hoping to, inspire open science practices through this and collaborative efforts.
And in true Twitter language, we are happy to report that someone has asked a question and now they're being advised to go to our website and our Git discussion. So hopefully this is live and please check it out. And yeah, that's really it. Thank you for your time.
Fantastic. Thank you so much. Now I forgot what the next one is. I think it is the neuropsychiatric brain states are the next ones up. Come on up. I see you're already, so you can share your. Oh yeah. Share screen. No, try sharing your entire desktop. You share desktop one, that one. Yeah. No.
Maybe I could send it to someone.
Yeah, you can send it to me.
Okay.
Let's maybe go to the next one, and it's the unseen, and then we'll come back to this one later. And it's the unseen. You're up. So one of you will need to be on Zoom and sharing your screen to present. Yeah, you got that. Okay, great. I'm going to put it here. And then what I suggest is that you look at the laptop while you're presenting. Okay.
Okay. Hi, everyone. I hope you're awake. I know these masks are so warm, behind them is so warm and cozy. But this is fun stuff. Again, on NSD project. So I think we are the 5th group maybe, talking about the NSD data set. We are specifically interested in motion perception and social cognition. And specifically, we are gonna look into a couple brain areas and see how these are and how and where these are stored inside our brains. So let me move to the next slide. As I mentioned, we are gonna use the NSD data set. We are interested in implied motion and social interactions. Let me show you some examples that come from NSD. So we see 3 pictures with different levels of motion. Maybe not so much motion happening in that picture on top. The woman is just sitting there. The one on the right,
is walking with some slow pace, and then we see higher levels of motion in this picture on the bottom. This is one way of looking at these images. Of course, we can ask other questions and label these images in different ways. So this is another thing we're interested in. We see on the top left one, maybe just one person skiing or doing some sport just on his own, and there is no much social context happening there. We can maybe look at the bottom right image and label that as kind of a general social interaction. Some of these pictures, we see joint attention in them, including, let's say, this one on the bottom left one. So these are two general ways that we look at these images and annotate them, which Satesha is going to talk more about that.
Okay, so as Sajjad said, we were interested in the basically the representations of these two sources of information in the brain. So the first one is whether basically the representation of social interactions in these pictures that all include people are basically distinct between these categories of like a single person in the image, multiple people in the image, but they are unrelated, whether they're attending to the same thing, so they have joint attention to something, or they are in an actual social interaction. So the sort of levels of these people getting engaged in a social interaction. And also the question about motion was whether basically there are graded representations of these different levels of motion that Sajjad showed basically different pictures for. And in order to do this, we're going to use the automatic and basically automatic coming from COCO dataset and manual annotations that we did ourselves of NST images. We're also going to basically use a multivariate pattern analysis on the task-related fMRI data from the NST, which include the classification analysis using SVM and also RSA analysis. And we're going to report the results, basically run the analysis on subject 1 in the native space of subject 1, although the MNI space is also provided. And we're going to run our analysis on not the whole brain, but two regions of interest of TPJ and MTL. And the TPJ mask we're using is in the MNI space, and we convert it to the native space, and the MTL is provided in the NST data in the native space. And so for the annotations, I said we used two sources of annotations. First, we started with the annotations that are provided in the COCO dataset. So these are all the categories of the images in the COCO dataset. We specifically chose the images that include person supercategory and animal supercategory, which involve like different animals, because we were interested in social interactions for people and also people and animals for motion. And then we took these images and we basically wrote a simple app in JSPsych, if you're familiar with it, to basically load these images and show it in a nice way for us to manually annotate. So we had like two small apps, one for motion. So you see the question is how much motion do you perceive in this picture, rated from no motion 1 to high motion 10. And also for the social interactions, what type of interaction do you see in the picture? So these are, for example, in a social interaction type of picture. Okay.
Okay, so we did some annotations manually. It took some time, but we have these labels. Let's start with these three different types of interaction. We have joint attention, social interaction, and unrelated people, as I showed you some examples in the first slide. This is the very bad chance results we get on this type of classification. As you see, we see the confusion matrix here, and all these three are more are being classified more towards unrelated people. It's really hard to study abstract concepts such as these social interactions or implied motion on these images. They go really fast. One important note is people fixate on a small red dot in the center of the picture, and maybe there are people or some joint attention happening on some part of the picture, which is not super visible when you fixate on these pictures. So that's one aspect of why we get chance accuracy. But the other thing is, as Satish mentioned, we are using some masks, including for this part of analysis, TPJ mask, which we take from an atlas. And in yesterday's breakout session on these atlases and primary visual areas, we saw the challenges of getting beautiful boundaries around, let's say, V1, V2. And you can imagine how hard that would be to get a mask, a functional ROI like TPJ from an atlas. And the ideal thing would be to have, let's say, some theory of mind localizer done on these subjects that participated in this study. So these functional masks are not really coming from the subjects. It's just something you take from an atlas. The other thing is, for some part of the annotations, we directly take things from the COCO data set. And this beautiful picture, one of the labels assigned to it is people. There are beautiful bananas here. And if you pay close attention, there is this man here, maybe munching on some banana or selling a banana. And this is one of the images that we kind of put under this people category. And you can imagine why the classification is not going right. So that's one of the future directions, to change these annotations and reclassify these different social interactions. Although this is not really meaningful, but I'm showing where this TPJ mask is located, and these voxels are showing the best voxels that are distinguishing between these three different types of social interaction. And if we got some high accuracy voxels, we would see maybe some light areas inside TPJ, but this is a chance, nothing really interesting happening here, but we are maybe bragging about visualizations here, although it's just 2 clicks in FSLIs. But anyways, another way of looking at things is, rather than just simple classification, we were interested to look into individual images and see how similar or different they are compared to each other, rather than just putting one SVM classification line between them. We are looking at all of them together, look how different they are. This is representational similarity analysis. We are looking at distances here. And it seems like there is something happening here. There are some of these images that are more similar to each other and are far from some other images. That's just one first step in RSA analysis. And then Satish is going to show you how we are going to deal with matrices like this.
Okay, so in order to see whether there is a structure in this matrix or not, we performed the hierarchical clustering on this matrix, which is here. So you see that, as Sajid was saying, there seems to be some clusters of like some images that are being clustered together, like a large group of images together, and some smaller clusters. And the next question we wanted to ask was that whether actually these clusters correspond to the categories that we're interested in the first place, in the social interaction annotations, basically. So now, in order for you to see it, I'm coloring these 3 different categories. So unrelated individuals, joint attention, and social interactions. And what we hope to see was that basically these clusters correspond to like 1 or like 2 groups of these types of like people images, whereas it doesn't seem so. So although there is some structure in TPJ in the NSD data, they don't seem to correspond to the social categories that we were interested in. But one other point that I want to note here is that this hierarchical clustering was done with the word linkage function, whereas like this is like one thing I want to highlight here, which is sort of in line with the multiverse analysis and a discussion about how different analysis might change your results. So we tried different linkage functions to see like how the clustering changes on this matrix. And although we didn't like dig into this more closely, it seems that the clustering would be affected by the linkage function that you use. So this is something that like for us that want to do like RSA analysis in the future, it's important like to think about and consider. But yeah.
Okay, the last thing we did is we drew these RDA matrices for motion also. What we've been showing is those three types of interaction, social interaction, and we put those images with different level of motion to see if there is any similarity between them. And this is what we get inside the MTL mask. There is just noise happening, it seems like, inside this mask. We also tested another mask of TPJ, which we talked about before. There seems to be maybe something happening here, and these are clustered, actually. This is after clustering. But even on TPJ, we are not seeing anything. It seems like noise. I talked about issues with these ROIs, and one future direction for us would be to, rather than using these ROIs, and as I'm mentioning here on my second point, is to run searchlight analysis. So we don't have these functional ROIs on these subjects. We cannot really bring them back. We didn't even collect the data. So one way to deal with this is to run whole brain searchlight and look around the whole brain, see if there is any interesting part of the brain representing these different social interactions or images with different levels of motion. Another future direction is, as I mentioned before, to re-annotate these images. We saw one particular example with images being labeled as people where there is no person involved in that picture. And all we showed was just one subject in this data set. We have 7 more, I think, and we're gonna try to do the analysis on all the subjects. And last but not least, I want to thank especially Ariel and Noah and others, and 3 exclamation marks to say how much we are thankful for that. So yeah, thank you so much.
Thank you. All right, and now to the last presentation of this marathon. Let me just share my screen here. I can share it now. Something is wrong with your computer, Jake, I think. Okay. I was just looking at this screen of your instead of the flat screen.
Okay, so the title of my project is Neuropsychiatric Brain States. It originated as ADHD brain states, but the reason we switched over was due to the data set that I was able to obtain and had more than one diagnosis. Therefore, we switched to neuropsychiatric brain states. I'd also like to quickly thank some collaborators at Mackenzie, Romina, Mishan, and Alice, who definitely helped out with some of the earlier stages of the project. And there is a link to the GitHub. So what is a brain state? Dynamic changes have been tracked from resting state functional MRI to when a person is conscious but not carrying out anything during the scan. And these diverse dynamical changes are believed to represent different internal states in the brain in terms of brain region interactions. So why do we want to study these? An elusive goal of computational neuroscience is to describe the brain as a dynamical system with a predictable natural temporal evolution in response to input. And this becomes pretty interesting when we consider psychiatric illness, because right now we really don't have any definitive neuromarkers that would indicate if someone has ADHD or if someone has depression. And this becomes a little problematic. So such models would be invaluable to clinicians for a generalizable tool for identifying optimal brain stimulation approaches to drive the brain from various states of disease to states of health. And this would be really important. But again, right now, we don't have anything like that that exists. So one of the questions that I had was, can we use brain states during resting state fMRI to accurately classify a diagnosis? So the specific data set that I use is the UCLA Consortium for Neuropsychiatric Phenomics, LA5C study. And there's a link if anyone is interested in looking at it. Using DataLat, it's actually really easy to obtain. And this study actually included 271 individuals. 3 were removed due to missing or incomplete data, but we have about 138 subjects who were healthy, meaning they were controls. We had 58 who had schizophrenia, 49 who had bipolar, and 45 who had ADHD. So walking you through the analysis pipeline here, using OpenFMRI, I was able to obtain the data set. Using DataLat, I was able to download it. And what I use is the YAO7 Atlas mask from NeLearn to actually obtain the brain states. And I was originally going to use voxel-wise analysis to obtain the states. The issue is that I ran into time constraints. Processing that much data just was going to take weeks on end. And unfortunately, my computing cluster wasn't working. So I shrink it down and use the YAO17 Atlas. And from there, what we do is we extract the time series for each parcel. And what I apply is spectral clustering to obtain 5 states. So you could use many different forms of clustering to obtain these states, and you can chain states. I used 5 just based on some of what the previous literature had indicated. And what I do is from there correlate states across subject. Now, this is probably one of the harder parts because basically what I'm assuming is that all my subjects are, 1, going to have 5 states, and 2, each state is going to be fairly similar to one another. So what I basically do is just take the max correlation for each state across individuals, because if you've ever worked with any clustering algorithm before, the unfortunate reality is that state 1 for subject 1 might be different than state 1 for subject 2. So that's what I'm doing with this analysis. And using XGBoost, I'm using a multi-class classification to determine how well I could use these states to classify a given diagnosis. And then finally, what I use is SHAP for feature importance to determine if, specific states are indicative of a diagnosis, or if time spent in a specific state would be indicative of a diagnosis. So let's look at state 1. I'm showing 2 different figures here. This top figure is called a multi-dimensional scaling plot. And basically what this is doing is it's taking the states across all the YAO17 networks and projecting it in a 3-dimensional space. So instead of viewing on a line plot at the bottom, basically what we'd be seeing here, if these diagnoses compared to controls differed in terms of brain state 1, we would see much farther separation. However, as you can see at the bottom plot and in our multi-dimensional scaling plot, that we really don't see any differences in brain state 1 across these diagnoses. Now, unfortunately, this is going to be a pretty consistent trend across all of our brain states, that what we're seeing is pretty similar states across all these diagnoses. Now, one of the interesting things for me was the first thing I thought of, well, maybe the data got processed incorrectly. However, the states actually do make sense, because if we look at this one right here, we could see that this state is particularly low in ventral attention A and ventral attention B. So someone might be a little more focused on this one compared to some of the other states. And all the diagnoses follow a similar trend compared to the controls, which I actually wasn't anticipating at first. But again, this is just the trend that we're seeing. And again, this is going to be consistent across state 3, state 4, and state 5. So as you can imagine, as we head into our classification, the first thing that we're going to find is that just using our individual brain states, that our classification is not that great. And if we try and classify controls, ADHD, bipolar, and schizophrenia in our multi-class classification, we're really no better than chance, which was a little unfortunate. So the next thing I wondered was, could we actually use time spent in a specific state to do that? Actually, I just want to show you this really quickly. So again, because our classification accuracy was really low, if we're going to look at our SHAP plots on the right here with the blue and red points, if we were going to see a specific state indicative of a diagnosis, what we would see is that either high separation with red dots or high separation with blue, basically saying that, okay, if someone was higher in a specific state, this would be more indicative of ADHD, for example. Unfortunately, we don't see that. And even if we look at our group average for identifying each diagnosis, we really don't get much separation. So this, again, led me to, okay, can we look at engagement or attention in a specific state? So now I'm basically calculating how long is a specific individual in a diagnosis spending in a state. And actually, the accuracy here is even worse. So again, this kind of leads to another question of, well, how reliable are these states per individual? And, you know, is there something going on with the correlations that are causing some of these issues? Because this is actually kind of a little bit different than what some of the previous literature has shown. It has shown that there should be a little bit of a higher signal when being able to classify or identify some of these differences. Unfortunately, I don't see that with either brain state or with time spent in a specific state. But again, for future directions, we can maybe look into some better results. But let's talk about some lessons learned, challenge faced, and some future directions that I'd like to go in. So I think from lessons learned, this was a great opportunity to work with NeLearn more and working with processing and, you know, just overall working with resting state data from the ground up, which is really great. And using Data Lab to obtain the data sets. This was the first time I've ever done this. So it was a fantastic opportunity that I'm definitely going to utilize more moving forward. Some of the challenges that I faced were obtaining multiple data sets. One of the reasons why I ended up using this data set to begin with was because it was the one that I had the easiest access to. There was other data sets that I was trying to obtain that just happened to be 11 years old that I wasn't able to access. So unfortunately, this was the only one I could use. And I think there still is something going on with the specific brain states across each of the diagnoses that could be impacting my results. And one of the other issues that I had was evaluating the specific brain states. I need to learn a little bit more and read up more on the literature about what each brain state means. And the last thing that I was running issues in, like I said before, was just time constraints with the voxels. There was like 40,000 voxels compared to 17. So as you can imagine, time was a little bit quicker with the Yale 17 atlas. And moving forward, I think I'd like to make comparisons between different atlases, not just the Yale 17, just using other ones as well, looking at these voxel-based analysis and looking at task-based. One thing with resting state is you kind of potentially say, well, everyone's just kind of fluctuating in the same states, but do we see differences during specific tasks that could be more indicative of classifying a diagnosis? I also talked about examining a multiverse analysis. Unfortunately, again, just time this week, I didn't have time to get to that. But I think different algorithms and different data sets would be really interesting to look at. And finally, testing a different number of states. I arbitrarily chose 5 because this is kind of an a priori analysis. But one of the big things that I was thinking about is it possible to take a data-driven approach? Is every individual in 5 states? Could they be in 2 states, 3 states? And figuring out ways that we could actually correlate these across subjects and maybe taking a more principled approach to identifying precise states for an individual. So that's it. I'd like to thank everyone who's helped me along the way. So it's been a great week.
Okay, and that brings our presentations to an end. Thank you, everyone. Just a few words of thanks here to, first of all, to the NIMH for funding NeurAcademy. And thank you to Noah and Molly, my co-organizers in organizing this week. And the TAs, I should have mentioned Mackenzie and Pierre, and Damien had to sort of disappear this week because it got very hectic. But Pierre, I think, hung around until the very end. And I think that is it. So with that, we will log off Zoom, thank our Zoom participants now, our online participants, and log off Zoom. And then for those of you who are here, we will meet in front of this building on Campus Parkway at 7 to travel to South Lake Union, or if you want to meet us there at 7:30 at the location where we will board the cruise at 7:30, and it leaves at 8. So if you're not there by 8, that's it. Yeah, thank you. See you all soon.